We'll jump into the second session. Gregory, please take it away.
Okay, thank you, Arie. So today, me, Ben, and David will be presenting our new algorithm for assigning types to neurons in the Connectome. And I'd like to start with a small apology that we're computer scientists and by no means biology or neuroscience experts. So the goal here is to take the Connectome and automatically assign types to neurons, something that is done mostly manually now. And furthermore, to do this, we will only use connectivity, so not morphology. We don't care about shapes of neurons. So we take the connectome and we treat it as a graph. The nodes will be the neurons, and the directed weighted edges are the connectivity, the synapses between the neurons. Furthermore, our algorithm will also take in a partial labeling. This is an optional thing, and I will talk about this in a bit. So here, for example, maybe we have experts that label these three neurons, and they have like exclamation marks. And these nodes with question marks are the neurons for which we want to get labels. So this connectome, or this graph, will be the input for our algorithm. And our algorithm will assign labels to the unlabeled nodes, unlabeled neurons. And this is how we can label neurons in the connectome. I should talk about this optional partial labeling thing. So you might think, if it's optional, why not just never use it? And here our algorithm breaks down into two versions, a seeded and unseeded version. Seeded is where you have these partial labelings. Unseeded is when you don't have them. And seeded is basically faster, much faster, and gets better results, but it does require these labels. Okay. So let me try to state these problems a bit more formally. For the seeded version of the algorithm, we have a graph with some partial labels, and our goal is to assign labels to unlabeled nodes such that the accuracy is maximized. There is a ground truth, and we want to get as many nodes right as possible. For the unseeded version, it's a bit more subtle. So the graph doesn't know about types, so it just groups nodes together. And the thing is that you can switch the groupings, right? If the graph says these nodes are all red, for example, and these ones are blue, you can switch blue and red. You can still talk about accuracy, but then you need to map the clusters returned by the algorithms to real type labels. And I don't go too much into the slides. It can be done efficiently. Okay, so this is what accuracy means here. You map the clusters returned by the algorithms to real labels in the best way possible, such that accuracy is maximized. A little bit about prior research. Perhaps most of you are familiar with NBLAST. So NBLAST is a tool that uses morphological data, that is shapes of neurons, to calculate a similarity score for pairs of neurons. And there are also some deep learning approaches that try to do basically what I described here. But they all use morphological data, and they don't really scale well to many types, neuron types, or very large data sets. Our algorithm is not a deep learning algorithm, and it's a very simple iterative classification algorithm, and it doesn't use any morphological data. Let me give you some of the results. So I'm going to focus on two regions in the fly brain. This is for the FlyWire dataset. There is the central brain, which has many types and few neurons per type. And there are the optic lobes, which have fewer types, but many neurons per type. I'm going to compare the seeded version of NTAC, the unseeded version of NTAC, and NBLAST. To make NBLAST a classification algorithm, you can combine it with a KNN classifier. So what you see here on top is the accuracy that we get. On the bottom, the gray bars are the percentage of labeled nodes used by the algorithm. And note that for the unseeded version, this is simply 0. It doesn't take any labeled nodes. So let's look at the results. So for the central brain, we perform about the same as NBLAST, although we're much faster because we don't really need to worry about morphological data. On the visual system, we perform really, really well. And this is actually very surprising because even the unseeded version, which has basically no labels, just looks at the structure, manages to get very high accuracy. So this is very interesting. And you will note that we have missing bars for the unseeded version for the central brain and the full brain. And this is because they are just very, like the number of types is very large, and we don't really scale well when the number of types is large for the unseeded version. Okay, and now to describe the seed algorithm, I'll give it over to Ben.
Hello. I'm just going to share my screen. Okay, is that visible and can you hear me?
Yes.
Okay, great. Okay, so thanks, Gregory. Thanks for having us, everyone. Okay, so I'll talk a little bit about our seeded algorithm. So as Gregory was saying, we start with some graph where we have these colors, which are these cell types that some experts have given us. In this example at the top, we have red, green, and blue. And the idea of this algorithm is that we first give all the nodes we don't have labels for the same color. So here we're just going to color everything we don't know red. And what our algorithm is going to do is it's going to run this iterative process where in the first part of a step, we take the partition of our graph, use the information about the connections between the neurons and the current partition to construct an embedding. That's visualized over here. And from this embedding, we're going to update our partition to get a new partition. And by changing the partition, we're changing this embedding. And if you repeat this process to convergence or just until you run out of your compute budgets, then you end up with a good partition, and that's what we return. So I need to explain how we build the embedding from our partition and then how we update the partition given an embedding. So let's start with the first one. So we're going to think about how to build an embedding for a single node, this white node, and we're just going to focus on a single color for now. And for the purposes of this embedding, the color of the node in question doesn't matter, and we ignore any self loops. So what we're seeing is these red nodes which have connections into our white node, and then these other red nodes at the bottom which have connections coming from our target node. And the way we start this is that we're going to compress this information into just two numbers. Oh yeah, yes. And for the purpose of this embedding, we don't care if we know the labels of the nodes coming in and going out for sure, or if we're unsure about them. It's oblivious to whether we know these labels or not. And the idea is that we're going to compress this information into just two numbers. So we have 6 connections or synapses coming in from this red type to our target node, and we have 9 connections going out to this red cell type from our target node. And we can repeat this process for all the colors or cell types we expect, and this gives us a bunch of numbers. If we have k cell types, then we're going to end up with 2k numbers. And we can think about this in two ways, either as a vector with 2k dimensions or as a histogram, where the first k components tell you how much connections are you getting from each of the cell types going in, and then the latter two tell you how many connections are you sending out to these cell types. And the intuition for why this process works at all is that in the brain, if you have 2 neurons in the same cell type, they should behave the same. And when I say behave, I mean they should talk to all the other cell types in a similar way, and they should listen to them in a similar way. And what that means is that if you're in the same cell type, then these histograms should look the same, and these vectors should be close. And we're going to use this property to go back from these embeddings to the updated partition. Okay, so how do we get an updated partition? So on the left, we have a graph where the exclamation marks are things we know for sure. These are from the experts, and the question marks are labels that we're not sure about. And we run this embedding process, and we get the embedding on the right. Another question is, how do we update the labels for the things we're not sure about? So we use quite a simple process. We just look at nearby nodes, and you assign the label of your node you're not sure about to be either the closest known label or some weighted k-neighbor majority. And to do this, we need some notion of distance. And so I'll talk a little about which distance we use. So it's quite a standard similarity. This one appears in the literature a lot, but we also tried others, and they're reasonably similar. So this is the Jacquard distance, and it's 1 minus the Jacquard similarity. And if you haven't seen this before, it's reasonably straightforward. If I have two histograms, then you can sort of lay them over on top of each other. And on the numerator, we just have the sums of the where they overlap at the bottom, so the minimums, and then on the bottom you have the sum of the maximums. And as you can imagine, if the histograms are similar, then this is very close to 1. And because the distance is 1 minus this, the distance is very small. So if something has got a small Jacquard distance, then the histograms are going to be very similar. Okay, so we have a way to go from a partition to the embedding, and then we can update the labels of the nodes we're not sure about in this embedding, and we get a new partition. And as we do this, because because these are coupled, as we update the partition, this embedding changes, it moves. And we do this either until convergence or until we reach the end of our compute budget. Okay, so at the end, after we run this for a bunch of steps and we converge, we get the outputs, and this is what we return. Okay, so this is a very straightforward algorithm, and we find that we don't need that many iterations. In the paper, we were doing 20 iterations, and this can be done very easily on a laptop. So now I'll go into a bit more of the experiments we did. So what you're looking at is plots using the FlyWire data where we vary the amount of ground truth data that we feed to our algorithm. These are the cell types of neurons we know for sure. And we make this assumption that we know at least one, we have at least one label for each cell type. Otherwise, when we do the majority voting, there's no chance that we'll assign a node to a cell type which doesn't have a single node in, or at least one node in. And as Gregory was mentioning, this method struggles more in the central brain. This is where there's lots of clusters with few neurons per cluster, but we do very well on the optic lobe and the projection neurons which connect it to the central brain. And here we get down to about using 8% in this graph, and we get very good accuracy for the optic lobes and projection neurons, and sort of okay-ish accuracy for the central brain. But if we focus on just the visual system, then we do very well. So we can go down to almost 1% and still get accuracies north of 90%. So this makes us very optimistic that we can use this method for other connectomes and get reasonable or very good accuracies. And the last thing I want to talk about is Arie asked if we could use this to try and label some of the neurons in the bank dataset as it's being labeled. And this is challenging, not just because there are many types missing when he asked us to do this, but also because the topology itself was incomplete. There were neurons missing, there were synapses missing, there were chunks of it missing, and this will have distorted those histograms or those vectors. So our method was struggling to deal with this, but we found some ways of dealing with this. So one technique we used was adding size constraints on the clusters, and this was informed based off cell types from FlyWire, which we could map over to bank. And this helped us label 5,000 or so neurons with some amounts of manual intervention over maybe 6 hours or so. Okay, so now I'll let David talk about the unseeded version.
Thank you, Ben. Let me share my screen. Okay. Do you see my screen?
Yes, you're good.
Okay, perfect. So as Gregory mentioned before, we had two versions of this algorithm. One of them is completely unseeded, so it requires no label information, which is what we're gonna describe now. the thing is, how can we generalize this algorithm that we just saw to handle actually no label information at all? So it turns out that for this, we need to think of the problem as, from a slightly different angle. We're going to think of it as a pure problem in graph theory, and let's see how we can deal with it. So the input is going to be just a weighted graph representing the connections between cells, and the number that represents the desired number of clusters that we want. The output is going to be a pure partition, a clustering. As we have seen before, we can think of a partition as a coloring of the vertices. And now, which property do we want our cluster to have? Ben has already hinted at the property that we want, and it's exactly that the nodes that have the same color should have the same embedding. Okay, so what's the embedding? The embedding is the degree counts, as we have seen before. So basically, for given any clustering and any vertex in our graph, we can define the embedding of this vertex according to this clustering as the collection of degree counts between that vertex and all of the clusters. Okay, so we have k clusters, we have 2k numbers representing edges in and out of V, from V to every possible cluster and from every possible cluster to V. What we want is that nodes that end up in the same cluster, that is neurons of the same type, they should have the same embedding. More of this later. Now, the good news about this is that if we actually think about this property, if we phrase it in such a strong way, in which we want nodes of the same color to have exactly the same embedding, then it turns out that this is a well-studied problem in graph theory. These are called equitable partitions, and they have been known since the 70s, and we have an efficient iterative algorithm that can find them, okay? So that's good news. Let's code it up and see if we can find the cell types, right? Unfortunately, life is not always that simple, we know. The main reason is that this condition is too strong. First of all, even if there was an exactly equitable partition that represents the cell types, any kind of noise in the data would destroy it. Second of all, it is too much to ask for exact equality of degree counts. Now, it is true that for any graph, you can find a perfectly equitable partition, but it will usually require an enormous larger of clusters. Usually it will require one cluster per vertex, in which case the problem is totally trivial. Of course, if I put a cluster per vertex, then the cluster will determine the degree counts, right? Because each cluster only has one vertex, but that's not an informative partition. So our main challenge is to actually relax this condition. We want an approximate version of this condition. So the input is the same, the output is the same, a clustering. The input is a graph on the number of clusters. We want to output a cluster. And now we want that vertices in the same cluster should have almost the same embedding, almost the same degree counts. Now, what does almost mean? Again, we're going to use Jacquard distance. For any given vertex, we can measure the distance between its embedding and the median embedding of all the vertices in this cluster. So I can look at the vertex here and all the 3 red vertices. I can look at the embeddings of these 3 vertices and compute some kind of average. And then I can see how far the embedding of this vertex is to the average of the whole cluster. If it's very close, then it means that it belongs to the right cluster somehow. If it's very different, then probably this vertex should be clustered in some other way. Now, once we can do this, we need a global objective function. Basically, what I just described is how to measure the distance between the embedding of a single vertex and the median embedding of a cluster. But we can sum all these costs for all vertices, right? So our goal is going to be to minimize the sum of Jacquard distances between the embedding of each vertex and the median embedding of each cluster. So what is the problem here? The main problem is a kind of circularity in this definition, because these embeddings are just degree counts. They depend on the partition chosen, right? So we cannot just say, okay, I know the degree of every vertex and I can just cluster according to the degree, because once you change the cluster of a vertex, you change the embedding vectors as well. So you need some kind of consistency property that allows us to find the right clustering. And this is actually what makes it hard to apply standard clustering algorithms such as k-means or modularity clustering or any of the non-clustering algorithms, because usually they assume fixed features, let's say, whereas our embeddings depend on the cluster itself. Now, the main idea here is that, okay, but what suppose we could have a good set of points that are representative of all the clusters. Like if I could identify a set of vertices and say, okay, this vertex behaves as a typical vertex of the first cluster. This vertex behaves as the typical vertex of the second cluster, and so on. Then we could run the seeded algorithm that Gregory and Ben just described, right? We could just take these good representatives as seeds, run the seeded algorithm, and then we would find a clustering. So the idea is to leverage this by building a clustering iteratively. We're going to start with everything into a single cluster. We're going to pick one vertex that is somehow is typical of the whole graph. And we're going to repeat this for a certain number of steps until we reach the desired number of clusters. What we need to do is to create a new cluster. How do we create a new cluster? Well, we need to assign a score to every node that tells us how good this node would be if we were to actually create a new cluster. Basically, we compute how much our objective function would improve if we made a new cluster with that new vertex. And we put it together with all the nodes that prefer to go with it in some sense, loosely speaking. Okay, once we have these scores, we're going to pick the highest score vertex. And this is going to be a new seed. So first, we start with a cluster into one single cluster. Then we pick the vertex with the highest score. That basically this score is telling us that this vertex is significantly different from all other vertices that we have. So it probably pays to start a new cluster here. So now we have two seeds. The seed that was representative of the whole graph and the newly found seed. Now we can write the seeded algorithm with k equals 2. The seeded algorithm will give us two clusters. Okay, now we have the red cluster and the blue cluster. And the idea is to continue this process. We need one additional technical step, which is basically, okay, we started with a vertex that was representative of the whole graph, but now we carved out some part of the graph into a second cluster. So maybe the best representative of the first cluster, the red points, now is a different vertex. So what we do is to recompute the representatives. Basically, every time we recompute, given the current clustering, we recompute a set of vertices that have embeddings very close to the median embeddings of the clusters. Okay. Then we compute the scores to see how to open a new cluster. We greedily open a new cluster by adding a new vertex to the list of seeds and running the seeded algorithm as a subroutine. So the seeded algorithm is a key component of this algorithm, but it can be taken as a black box. There's an overall strategy that works iteratively by creating new clusters, finding when to open new clusters, what is the best vertex to use that for. And then you call the seeded algorithm here. Okay, so this concludes the overall high-level description of the algorithm. If you want to more about the nitty-gritty details, of course, we'll be happy to answer questions about this. Let me just briefly mention some of our experimental results. So first, since this is, to the best of our knowledge, the first algorithm to detect cell types automatically, there's no real good baseline to compare it to. But okay, we chose as a baseline for the unseeded algorithm the following method. Basically, it's a kernel k-means using the similarity matrix computed from n-plus scores. So you can see actually that this doesn't really work very well. We can have here the accuracy, and we never really get much better than 7% accuracy using the baseline. And actually, it's interesting to note that even the accuracy does not always increase when you increase the number of clusters. And what this plot shows actually is also the objective function of k-means. Clearly, k-means is designed in such a way that increasing number of clusters should decrease the objective function. So we can see here that even though we are decreasing the k-means objective function, we're not really improving accuracy. Okay, this is the baseline. What do we get from our algorithm? So we depicted here the experiments for the main visual system and the old intrinsic systems. And what we can see here is that we can reach accuracy above 80%, totally unseeded. Let me just say, okay, here this guy's a speed up version of the algorithm that doesn't consider all possible seeds, but this is for efficiency purposes. If we run the whole algorithm, we get the same result, it's just lower. What's interesting to note here is that if you look at how the accuracy improves with the number of clusters and time, basically it reaches a point in which adding more clusters doesn't really improve anymore. So basically we can, in about 12 minutes, we can reach an accuracy of 83%. And from then on, the algorithm keeps adding clusters because we're asking it to, but the accuracy doesn't really improve. Another interesting thing here, because here, okay, we plotted accuracy and F1 score, but this line is also interesting. This is our objective function, the thing that we're minimizing. And it's interesting to note that the reference solution has a very low value of this, okay? This is not always the case, but it actually is also an indicative that our objective function is aligned with finding the ground truth. Okay, there are lots of open questions about how we could continue. As far as the unseeded algorithm goes, one first question would be how to improve the accuracy and the speed. It's not particularly fast because we were mainly concerned with finding something that works. It's not obvious at all that you can actually infer connectivity just from infer cell types and from connectivity. So our main concern is to find something that works, and then we didn't worry too much about speed. I'm sure they can be improved though, but it remains to be done. Another question is how to detect the best number of clusters automatically. Here we see that there's a point at which there doesn't seem to improve much if we go beyond that. So maybe there's a way of detecting the number of clusters automatically. I should say that if we actually increase this a lot, up to the total number of clusters, what we would see is that the blue line would go to 0. So it's not like you can just find a minimum of the global objective function, but maybe there's some way to determine automatically when it doesn't just make sense to continue adding clusters. Okay, and this would get rid of the only parameter that our algorithm has. The last point is for us computer scientists, it will also be good to study the complexity of this problem from a theoretical viewpoint, find approximation algorithms or hardware results. And as for the seeded algorithm, one of the key points is to see whether we could improve accuracy using machine learning techniques. Let me also say finally that I believe that this algorithm, since it provides a new partition and primitive, it could also have applications to other problems, both in neuroscience and in data mining in general. Maybe it can be used to initialize GNNs, for example, or something like this. It's a new kind of partition that has barely been studied before, and I'm confident that we could find more applications of it. There's a paper we have uploaded to the B archive, so if you're interested, you can have a look at it. Okay, this concludes my presentation. Let me know if you guys have any questions.
Thank you, David, Gregory, and Ben.
Come again?
Are there any questions for one? Wayne?
So at one point, one of you mentioned running some algorithm, I forgot exactly which one it was, until you run out of compute resources. What amount of compute resources were you talking about? Like, are we talking hours, days of CPU, or what?
I was being optimistic. I mean, the experiments you ran on the full brain for the fly, it took 10, 20 minutes to run on a laptop. But obviously, we hope in the future we get to run this on much bigger connectomes.
There was a question, Sujit, yeah?
Oh, yeah, so, you guys, so from what I understand, you guys have like a pure computational method, like no ML right now. And you mentioned that the literature currently requires, morphology for the current deep learning methods. But there's actually a technique in geometric deep learning where you want to label K types to these nodes. And like, for instance, if you have a 3D mesh or something and you want to label parts of that mesh that are like corresponding to different functions or whatever, you could use like some kind of geometric deep learning like GNNs, GCNs with K number of attention heads. And that would be completely, you wouldn't need any seeds either. So, and it would be I think it would still need to be supervised, but I just wanted to point that out in case that would be a future research direction as well.
Yeah, I think the second reference that I believe it was Gregory showed, the recent work, is using those, using the morphologies and in particular point clouds of these neurons to exactly generate these morphology-based features and cluster them. I believe, I forgot what Gregory mentioned, but I think the performance of those in terms of accuracy is lower than what we achieve here. But it's also a very interesting work to look into. It's very recent. It's called point affinity based. In fact, Mala pointed it out. Yeah. All right, maybe one more question and then we can move.
Yeah, so like, are the cell types, do they need all of the connections in order to define them? Or do the types end up being defined based on just like a few strong connections? Like if you prune away all the connections, do you get the same wavelength?
Were you able to hear the question? So to repeat, Duncan's question was whether you need all the connections in order to define the cell type or whether partial connectivity information is sufficient.
Yeah, maybe I can take this one. From our experience, looking at subgraphs or removing connections really hurt the algorithm. But I think what you're suggesting is removing weak connections or some smart way of removing connections. And we didn't really look at it. But labeling bank with partial connections or just subsampling nodes or edges was very detrimental to our method.
Okay, thanks again. And next presentation is by Dániel Barabási. Dániel, are you online?
Yeah, I think I should be screen sharing as well.
Yes, thank you. Perfect.
Okay, cool. So yeah, the title of my talk is Physical Network Constraints Define the Brain's Connectome. We have a preprint under a similar name. So if at any point or afterwards you want more information, feel free to check it out. So as a physicist, the brain connectome stands as one of the last, terra incognita for network science. And I think what I like about working on the brain and working on brain connectivity is that it's a case not just where physicists are trying to fit, spherical cows onto something that perhaps does not need it, but is a case where the kind of biological insight and the way that people were thinking about this from a neuroscience perspective actually matches in a very nice way the toolkit of network science and of physics. And on top of this, I mean, there's a lot of development for network science that is necessary in order to make this all possible. So I think there's a lot of work to be done in the future, and hopefully some of that is a start that we're showing here. So, the initial idea of neurons as networks, or one of the kind of major steps towards thinking of this way was with Roman and Gahal with these early imaging steps. And it took almost 100 years to get the first kind of connectivity diagram, the actual network. And that was in C. elegans, right? And C. elegans was kind of our bread and butter as network neuroscientists for a long time of looking at cellular level connectomes. Of course, this was all until last year, where the amount and availability of connectome data absolutely exploded. So of course, we're here to celebrate FlyWire, and this is in many ways, the jewel of the crown of these many different data sets. And so this came out last year, and it allows us now to kind of a full or rather complete central nervous system for not just for the fly, but for the larva and C. elegans, as well as some kind of partial or ventral neural cord for the fly as well. And then there's also some partial data sets from the zebrafish the mouse and the human, two of which came out in the last year as well, right? So looking at all these different data sets, we became excited by the possibility of understanding the network features of the different data sets and seeing what sort of kind of conserved properties, as well as like diverging properties you might see across both many different organisms, as well as many different perhaps screen like captures of the same organism as we might have for the fly. So as we went about this, the first thing that we often study is the node degree, which in more neuroscientific terms is that for each node would be a neuron. A degree is the number of distinct neurons it synapses with. So we're counting the total number of partners here. And what we did is we just measured this for every single organism or every single connectome we had access to. And what we found is that there seemed to be this reproducible, we seem to see the same curve over and over again when looking at these different data sets, right? And we started, but the main thing that we saw is that these curves didn't really match the things that we would expect from a network science toolkit. And just as a quick background, the way to read these curves is we're writing the probability of having a certain number of partners on the y-axis. On the x-axis is the number of partners. And here we're doing log binning. So we're grouping different neurons into like, log levels. So this is 10, 100, 1,000, 10,000. And we'll notice that there's a peak in C. elegans around 10, but then the peak in mouse and human is on the order of like 100, right? So many different neurons, a large number of neurons are found within each of these points. And this would normally be a histogram. It's just nicer to show these points. Okay. And this is all to say that, we find this very repeating pattern, but it's not really familiar to us from a network science standpoint. Specifically in network science, there are two major classes of degree distributions that we tend to look at. The first one would be random networks, where you're basically saying for the Arie you would be like, you have a bunch of neurons and a neuron can maybe make 10 partners, and it just randomly picks 10 neurons to make partnerships with, right? And this leads to a Poisson distribution, which looks like this on a log-normal plot. And then the other one that we see is that is a kind of a growth-based model where you're constantly adding in new neurons, and they can pick from any of the existing ones. So it's kind of like you're kind of adding on ones continuously, but they do a random choice. And this leads to an exponential distribution. The other one is a more structured distribution that you might find, which is with growth and preferential attachment. This is what you see in the internet or technological networks often. When new websites or neurons come in, they'll pick from the existing set of neurons, but they tend to pick the most popular neuron or the most highly connected neuron available. This is like how most websites will connect to Google or Facebook or one of these large websites. And real networks are often characterized by non-trivial combinations of these elements, or there'll be noisy versions of these, but normally you can fit one of these distributions to a good extent. And in the past, we've looked at, or people have looked at C. elegans and compared it to randomly growing network model. So the C. elegans cellular connectivity, and as well as in the brain and fMRI networks, which are of course a completely different scale, but people have shown that this has a scale-free property, right? So there's reason to expect that we would see one of these distributions. But again, when we looked at node degree, it seemed to be none of these. So in order to just kind of validate our first point that these distributions look pretty similar to at least to each other before validating what they are, we decided to rescale them. And basically what this means is that for each of these distributions, we'll subtract the mean of the distribution and divide by the standard deviation. So just showing the mean of this, right, as I mentioned earlier, there's a nice basically gut check that as we go to like larger organisms or larger connectomes as well, you'll get a higher mean. So larger brains have more numbers of partners of neurons on average, right? You get the standard deviation, and then you rescale, right? And again, now this all looks like it's falling onto the same distribution. And indeed, when we fit a distribution to this, we find that a single distribution seems to characterize all of these different organisms. Of course, just with a different mean. So, kind of unveiling what distribution this is, we find that the node degree is fit by a log normal distribution with different means per connectome, right? right? So it's the same distribution, you'll just have, you know, a different mean value across these values. And these lognormals are interesting because they provide a diversity of neurons available. Most of these neurons will have degree near the mean, like a very, very large amount of them, but there also exist long tails in both directions, similar to what like a scale-free network would look like. Allow for like you to have both low degree and high degree neurons readily be available within the data set. And this, you know, this has like computational implications. You know, it's like most of your neurons have a certain average value, but you can still have these super connectors or really sparse connectors within the data set, which you can use to kind of either integrate information or just have very specialized input patterns throughout this. So we started to ask then, like, where is this coming from? So a big part of network science is that, you know, we have an explanation for where these data sets come from and like why a certain distribution comes about. As I mentioned, random connectivity or preferential attachment, something like this. And our hypothesis was like, okay, node degree comes from the number of synapses neurons have. Maybe there's some interesting correlation there that we could find. So we looked at node strength, which is essentially just synapse number in this case, where we're adding up the total number of synapses that neurons make. And again, we find a very similar distribution, and we can play this game where we can rescale it, and we find that they all kind of fall onto the same distribution again. So now we have two different distributions that have the log normal pattern, and we don't seem to be any closer to an answer as to what's going on under the hood, despite having the statistical characterization. And I mentioned that the reason that we like, you know, existing distributions like these random networks or the scale-free networks is because we have an explanation of where they can come from. If you can find a biological mechanism by which neurons can choose, you know, a random connection, or by the fact that they're growing and choosing random available connections or random available neurons along the way, then you could, you know, connect the kind of the statistical pattern that you see to the underlying biology that you find. But log normals have been extremely undercharacterized, even at a theoretical level within network science. You don't see a lot of distributions like this, and none of the models, or maybe 2 or 3 papers that you might see related to it, they're not related to biology. So we started to ask, how can this actually come about? And to do so, we started to reevaluate how we've been looking at the network so far. So as network scientists, you know, we like having the simplified version where we look at, you know, the great complexity of the brain as a number of different nodes with various amounts of various strength of connections between them, right? But of course, the connectome is a physical network. These nodes and neurons are physical objects that also serve as the links. So in order to make 30 different synapses, you need to have an R bridge, an axonal or dendritic R bridge, long enough to be able to actually support 30 different connections. And, you know, for each of these neurons, we also have labels, as many people have talked about in previous talks today, where you can get both, you know, the degree, so the number of partners, the number of synapses, but also the length of these neurons, you know, their neurotransmitter profile, as well as their the density of connections along them, right? So we can get these for all different cells. And we wanted to look at the physicality and see from the physicality whether or not we can derive why we're seeing the sort of distributions that we've observed that had previously been observed in largely unobserved in network science. So to do this, again, we looked at the physicality of neurons, and we said, okay, understanding this kind of 3D architecture is extremely complex. We do need some sort of simplifying assumption. And we just said that each neuron is an incomplete tree, right? So you can represent this neuron that's spinning over here as this tree structure that we have over here. And the only reason it's incomplete is that it's not like a perfectly balanced tree. You're not making a double kind of division every single time you make a division, but you see that, you know, you might have certain, let's say, dendritic arborages that stop early, whereas other ones run later, right? So there's this kind of lack of balance, but this is probably important for, you know, for the connectivity structure and for the integration structure in this case. And the nice thing is that this sort of incomplete tree has been well characterized by a Watson-Galton process, where it was used to kind of model like family lineages and the survival of family lineages. But you're basically saying that, you know, every kind of division step, there's a probability of survival of, and, you know, probability of branching that you can have here. And there's also a probability of not branching, therefore, right? And if you run this simulation for extended periods, you'll end up getting these sort of incomplete trees that you see on the right-hand side. And there's very nice derivations for the length of the surviving branch of just like how long then is the longest branch. But you'll have to add this up over all the different branches that you have along the way to get the full length of a neuron. The nice thing about this is that this is a multiplicative process. So this kind of repeated branching, re-rolling process is multiplicative, which has often been linked to log normals in the past. So we wanted to solidify this connection and see if this could be the underlying function that leads to the log normals. And an important part of this is thinking about a critical value of survival of 1/2. If you're at 1/2, then you have 50-50% probability of creating a branch. Then each time you're creating 2 new nodes with the branch. So it's like 2 times 1/2 gives you this kind of critical value of continuation. And in the subcritical regime, when you're actually below this value, right, you're more likely to be ending the branching than actually continuing it, given that your probability of survival is less than the number of branches you're making every single time. This is a multiplicative process with no close and no close solution, but there's strong numerical evidence for a log normal length distribution of the resulting trees. In the supercritical phase, where you're actually branching more than you're kind of continually branching and you're actually very rarely stopping the branching, you also get a log normal distribution of neuronal lengths. So then we actually went out to measure the neuron lengths, and we again found that they're very nicely log normal. So we now kind of have this process where we can go from the kind of generative process of how a neuron potentially starts to grow, these rules of continuing to branch and kind of adding on branches of neurons. And we find that it leads to the length distribution being log normal. There's actually a lot of cool tricks that if you go to the paper as to actually how this works, because we find that it's not like there's not a static value of survival for every neuron, but that value actually starts very high and decays over time as the neuron branches. So it's like, you have a high probability of branching close to the soma, but then the further out, if you're deep into the dendritic tree, then the value actually falls a lot below 1/2. And you can use these curves to characterize different cell types and things like this. And they have, it's something of further study for sure there. Okay, but we got to this point where we have a length distribution that's log normal. And we wanted to get to a point where we can predict from the length distribution the number of synapses that neurons have. And the basic hypothesis, which is absolutely not a new one, is that the longer a neuron is, the more opportunities it has to form synapses with other neurons, right? So basically saying that the number of synapses you have is proportional to the length of the neuron, as well as the density of synapses for that neuron. And this value seems to hold up across all these different data sets, or this relation seems to hold up. And we can get these values, get these correlation values out of here in order to understand this. But the important part of this is that the length distribution and the density distribution are covariates for the number of synapses you make, and eventually the number of partners you make. And what is, we need to first look at the nature of the length distribution and this density distribution. We again find that these are both log normally distributed, as I had shown you already for length, but this is also true for kind of a local synapse density measure that we calculate. And therefore, we can make this kind of derivation, which is basically just saying, like, if these two values are log normal and you're multiplying them together, then the resultant will also be a log normal. And you can derive the synapse distributions, the number of synapses that neurons have, from the distribution of their lengths and the density of synapses that you have. And you could derive both the mu value for a log normal and the sigma value for a log normal like this. And so we kind of showed how well this works here, where in big letters we have to always write here that the curves are not fits, but they're actually the analytically predicted values based off of the length distributions and the density distributions. So the synapse distribution for zebrafish, for the fly, and the mouse, it seems almost perfectly captured by, like, it looks like you actually fitted the data by the analytical prediction that we make from the length distribution that we were able to predict from this growth process. So we're able to go from growth to length distribution to synapse distribution. And next, we want to predict the number of partners that neurons have as a function of the number of synapses that they make. And the basic statement that we make here is that a neuron's ability to acquire synapses is sublinear, right? Is if you have a certain number, if you have 10 synapses, right, you can make at most 10 connections. But many of the cases, you might be making, 6 connections, but with a few of those partners or one of those partners, you'll make many synapses. So that's all that we're stating here. We can find this, we can measure this across all these different data sets again, and we can see that across different species, there's a different level of correlation, where in the fly, it's between kind of like 0.7 and 0.8. And then for the mouse, it's actually much higher, meaning that there's like, you're actually making, you're more likely to make a single synapse with your partners, whereas like in fly, you might be making multiple synapses or kind of having more multi-synapse connections with your partners. So again, we can have this derivation. It's actually very nice that with this sort of relation, you can relate the log normal distribution of the degree to the log normal distribution of your synapses through this these fit values as well as the values underlying the synapse distribution. And again, we can make these plots where we're saying like, okay, these curves are not the fits, but they're the analytically predicted values of the degree distribution. Now, depending on, this should be the synapse values here. So mu s and sigma s. So the synapse values very, very nicely predict the degree values that you see here. Okay. So the full picture, right? So we can start from the physicality of the system. We can look at a neuron and we can look at its physical tree. We can take it kind of like an SWC version of it, so a simplified tree version of it, of how these neurons kind of turn into branching processes. And we can model this branching process using the Galton-Walton process. And we can use this to predict the log normal distribution that underlies neuron lengths, right? So this physicality leads to the neuron length predictions. And then we can use further observations about physicality to say that neuron length combined with the synapse density of neurons leads to the synapse number. And synapse number can help us predict the neuron degree or the number of partners that neurons have. So this is kind of like a nice full picture of how we can use the physicality of start to understanding the underlying distributions that we see within the brain. And as kind of like a final note of relevance, that again is something that we need to further explore, but given that this is FlyWire group that we should think about, is that these log normal distributions extend to cell types as well. So the single log normal distribution that we see for the entire fly brain is actually made up of a bunch of smaller log normal distributions of the different cell types. And these cell types will all individually have different means. And I kind of think of it of like, they kind of tile the overall log normal that you observe, right? So there's different generating functions for different cell types that lead to still this log normal distribution underlying everything in neuron lengths and the strength of connections. Okay. Other than this, why do we care? Are log normal networks any different from networks that we've seen previously? So one of the things that we often look at is the max number of connections that you can have or that you can predict within these systems versus the brain size or number of neurons. And we find a different scaling across random networks like the Poisson, the scale-free, which is this preferential attachment, and the log normal that we have here. And one observation of this is that in Purkinje cells, the number of synapses that they have is 200,000, but their degree is 7,600, right? In a scale-free human brain, with 10 to the 11th neurons approximately, would say that the maximum, but the superconnector neuron within the brain would have 10 million connections, which is absurdly large. However, a log normal connectome predicts, given that you have 10 to the 11th neurons within the human brain, roughly that you'd be on the 1,000 order for the largest connection, largest connecting neuron within the brain. So the predictions that we get are much closer, even with these very, very coarse fits to the observed values within the human brain. Another measure that I'm not going to go into too much detail that I often care about is this degree heterogeneity measure. We could derive these values as well. And very interesting is that like the robustness and the synchronizability of the brain is related to this measure. So this is like related to if you start taking out neurons, like in terms of decay, as well as how quickly the whole system can come together in a dynamic setting. And for log-normal networks, it does not depend on the size of the brain. So the neuron, or the number of neurons within the brain, is actually not present within this equation, where it is present in scale-free, which could lead to very kind of useful metrics related to kind of local connectivity and not having, or kind of different compensatory mechanisms that you might have to imagine for how you control kind of the spreading of information within the brain. So, okay. In summary, I showed you that the degree distribution, the number of synapses, and the length distribution within the brain are best approximated by log-normal distributions. And if we think of the connectome as a physical network, it allows us an analytical derivation of these values. We can see how the degree distribution comes from the number of synapses, and we can also see that how the number of synapses comes from the length distribution, and we can derive the origin of the multiplicative process, this branching process that leads to the log-normal length distribution. You know, there's nice falsifiable dependencies between the parameters characterizing neurons. We could start looking at like what the biology of these generating functions are. We can link it to many other models of neuron growth. We also have nice things related to rich club behavior, which is, I think, important to talk about because log-normal networks are a completely different class. And it means that when you're creating like dynamical models of the brain, if you're trying to create networks that you then simulate, you need to have the right underlying connectivity structure. So I can also talk about what this would be, but basically you want the right randomization, or you want to be using a log-normal as your baseline, not a random connectivity. And we find that the rich club behavior that many people study is actually comes from just the density of the system as well as the log-normal nature of the system. It then, not trivially, but like it rather easily predicts the rich club behavior every single time for the brain connectivity. And so overall, the log-normal connectome presents a novel network class where there's a lot of homework for network scientists to start looking at this network and understanding its properties. And also it leads to kind of interesting things like that have been studied by Bujaki and others related to kind of the computational properties of log-normal connectivity and log-normal activations within the system that need kind of further characterization from a neuroscience standpoint. So finally, acknowledgements. I'm funded by the Schmidt Center and by Future House. And Ben Piazza and I were co-first authors on this. And Andre, Julia, and Lasse also had a huge contribution on bringing all of this together. Thanks so much.
Thank you, Daniel. Are there any questions in person or online for Daniel? Yes, Alex.
Thank you. That was a very interesting talk, and I was, excited to hear about how you're fusing the physicality of the brain with network science. I was particularly interested in what can be done breaking this down to the cell type level, looking at the log-normal distributions of all these different metrics there. I wondered whether you'd considered or experimented with breaking these neurons down into their axons and their dendrites, because the rules for branching there can be quite different. Often, the axons versus the dendrite often define a lot of the input-output structure of given neurons or cell types. I just wonder how that could be incorporated into your workflow here, whether you've already tried to experiment with that sort of thing.
Yeah, I think okay. There's kind of the cop out, which is like we wanted to look at very universal distributions, and this becomes very hard with axon and dendrites, given how different they are in their annotation between invertebrates and vertebrates, right? So it's like while there is annotation in the FlyWire for axon versus dendrite, it's like not as clear of a distinction as like the canonical axon that you might see in like a mouse pyramidal cell or many like vertebrate systems. So it became a little bit harder to kind of lump them together and analyze them. We did analyze them separately, and if I recall, we do see a lot of the same properties. And we're also looking at this deeper, but more focused on the mouse side now, where we're trying to kind of, at the very least, make sure that these larger distributions that we see are the same across all of them, and then trying to fit this branching process, if possible, onto those data sets as well. So it's like, yeah, I absolutely recognize that it's different processes, but like kind of in initial investigations, we do see that at the very least, the length distribution of axons and dendrites both still follow log normal independently, they'll just be different from the means, and like, you know, maybe the parameters will be different from looking at the full neuron because you're cutting neurons into partial segments. So.
Thanks, yes, Wayne, and then after that we'll maybe take the rest of the questions offline.
Good talk, Daniel. Thank you. Have you considered using graphic analysis for the Connectome?
Graphical analysis?
Graphlet.
No, we have not, but now I'll look up in more detail what that means. If you have a suggestion, I mean, quickly, I'm happy to hear it, but
I'll send you an email.
Okay, great, thanks. Yeah.
All right, so 2-minute break just to make sure people are still focused, and then we'll do the third and final session. So we'll have maybe 10-minute delay in the schedule. Thanks, Daniel.